Welcome back, everybody, to the immune system. Today's submodule is on the immunoglobulins, so let's go ahead and get started. So activated B lymphocytes and the B cell receptors. Activated B cells proliferate and transform into memory B cells and plasma cells. B lymphocytes, like T lymphocytes, also have unique receptor complexes. There's roughly about 100,000 per cell. Each complex binds to a specific antigen. We call these BCR for short, and this is an antigen receptor of the B lymphocyte. B lymphocytes make direct contact with the antigen, and they need to contact the helper T cells, particularly the Th2 subtype, to produce a full activation. Plasma cells are the cells that will then release the antibodies. Activation of a B lymphocyte. So as with T lymphocytes, two signals are also required for the activation. However, the B cells do not require antigen presentation by other non-lymphocyte cells. B lymphocytes recognize and respond to antigens outside of cells, such as antigens of viruses, bacteriotoxins, and even yeast spores. The first activity and first stimulation of B lymphocyte occurs when an antigen binds to the BCR. This cross-link of the BCR is then engulfed and subsequently presented on the MHC class II molecule of the B lymphocyte. That will then be recognized by the helper T cell. The second stimulation of the B lymphocyte occurs when receptors of the B cell interact with receptors on the helper T cell, and the helper T cell releases another chemical called IL-4, interleukin 4s, to stimulate B naive lymphocytes. When these naive B lymphocytes are activated, they begin to proliferate and differentiate into two types of cells, the plasma cells and the memory B lymphocytes. Plasma cells start to lose their BCR receptors roughly about 5 to 7 days after they survive, and they begin to release a large quantity of antibodies to their environments. Whereas the memory B cells could theoretically live in the system for months to even years. Over here is a visual showing you the first signal. The free antigen binds to the BCR. The B lymphocyte engulfs it and presents it as an antigen to the helper T cell. Now, when you show it here, you can see the MHC class II co-receptor with the antigen is presented, and now it interacts with the B cell, the naive B cell. The IL-4 released from the helper T cell stimulates the B lymphocytes to then activate into the memory form, as well as differentiate into the activated form of the plasma cell. The plasma cells will then release antibodies that will then flood the body to help get rid of the antigens. The immunoglobulin molecule is a Y-shaped soluble protein that's composed of two heavy polypeptide chains and two polypeptide light chains that are linked together by a disulfide bond. And they form this antibody monomer. On the right side, you can see antibodies look like a Y-shaped protein. There's a hinge region. These S represent the sulfide bridges. And you can see that there's an FAB region, and there's also going to be an FC region. The FAB region is going to be the site that binds to the antigen. This binds via very weak intermolecular forces like hydrogen bonds, ionic bonds, hydrophobic interactions. Whereas the FC region, which stands for the constant region, is a portion of the antibody that determines the biological function of that antibody. It remains constant. It determines the kind of class that antibody falls into. The two long chains are heavy chains, and there is a hinge region which makes it flexible. Each of these heavy chains also have subsegments called the V, J, D, and C segment. These different segments are coded by different genes for different expressions, and that's why a chunk of our genetic code is for variations of antibodies. So most likely you encounter something in your lifetime, you will have the ability to create an antibody for it. The light chains are also subdivided into the V, J, and C segments, and these are linked together, as I said before, with the disulfide bridges. So how do antibodies recognize so many antigens? Well, it's based on the recombination of these V, D, and J regions. V, D, J regions represents different genetic combinations. There's 3 segments of this gene, each segment having 3 subunits. You can then randomize each gene fragment to create roughly about 10 to 14 possible variations of the light chain and the heavy chain. This will then encompass the majority of potential antigens you will meet in your lifetime. Each of these regions is made up of a small gene segment with multiple copies, and these segments are combined in random fashion so that you have a number of antibodies generated, and you go through a screening process before the final antibody is retained. This is another cartoon visual of the antibody structure, showing you the hinge region, the disulfide bond, the arm region, the variable region, and then the Fc region, the constant stem region. Actions of the antibodies. The FAB portion has these three functions. First of all, neutralization occurs when an antibody physically covers over the antigenic determinant of a pathogen and then renders it completely ineffective. Agglutination, which is clumping. This occurs when antibodies cross-link with antigens of foreign cells, causing them to physically clump together. As a result of that, precipitation occurs, which is when antibodies cross-link soluble antigens in the blood, causing them to become insoluble and then precipitate those out of the solution. The Fc portion actions include the following. We have the complement fixation, which occurs at the Fc region and activates the complement system, which is the classic cascade. This is a series of proteins, about 9 proteins that fuse onto the membrane of a target to cause it to lyse and burst open. This is triggered by the IgG and IgM antibodies. Opsonization. This occurs when the Fc portion of the antibody makes the antigen much more attractive to phagocytic cells and more aggressive. And then the activation of NK cells also occurs. These are called natural killer cells. They're triggered by IgGs, which could then release cytotoxins to kill off bad cells. This process is called the antibody-dependent cell-mediated cytotoxicity, ADCC. The classes of immunoglobulins. There are 5 subclasses of antibodies. They're called the IgG, IgM, IgA, IgD, and IgE, also known as GAMED, G-A-M-E-D. The first class is called the IgGs. These guys make up about 75 to 85% of all of the antibodies found inside your lymph, cerebral spinal fluid, serous fluid, peritoneal fluid, and this one does have the ability to cross the placenta. IgMs are found predominantly inside the blood. They exist in a pentamer structure, which means there's actually 5 of them stemming from the Fc portion. And these are responsible for blood type rejections when you have mismatched blood. IgAs are found in areas that are exposed to the environment, particularly in the mucus, saliva, tears, breast milk. These are expressed as a dimer. So there's 2 of these proteins linked together, and thus they're quite effective in agglutination. IgDs function specifically as antigen-specific B-lymphocyte receptors, and these are expressed as monomers, and they have a role to help immature B cells to activate. IgEs are usually formed in response to parasitic infections as well as allergic responses. They will attract eosinophils, and they're generally really low in synthesis in the blood. So if you have an abnormally high amount of IgEs, that could be indicative of a basophil activity, an allergic response, or anaphylactic shock situation. Class switching occurs when the plasma cells change the types of antibodies it produces. This does require the ability of a helper T cell and the plasma cell to be involved. We will not cover the details of class switching. On the right-hand side, you can see the different shapes of the antibodies. IgGs, IgEs, and IgDs are expressed as monomers. IgAs are dimers. IgMs are pentamers. Over here is a chart that shows you the size difference of each of these antibodies. You do not need to know these numbers, but it is interesting to show and compare. You can see clearly that the size difference when compare IgM to an IgG or an IgD or IgE. And then notice over here is the half-life, the duration that it lasts inside the body. The IgGs are very long-lasting, 23 days, whereas the other ones, IgMs, IgDs, IgEs, are very short-lived. Of all of these, only the IgGs are able to cross the placenta, and IgGs as well as IgMs are able to activate the complement system, whereas the IgAs, Ds, and Es cannot do that.